자, 김동훈 총장님과 프랑크 박사님께서 지금 입장을 하고 계십니다. 여러분 뜨거운 박수로 환영해 주시기 바랍니다. 안녕하세요. 저는 제3회 고려대학교 넥스트 인텔리전스 포럼의 사회를 맡은 고려대학교 이과대학장 양성덕 교수라고 합니다. 먼저 총장님의 환영사가 있겠습니다. 자, 뜨거운 박수로 총장님을 맞이해 주시기 바랍니다.
자, 오늘 이 토요일이고 또 어제가 제가 듣기로는 BK21 지원서 마감일이어서 많은 분들이 밤을 샜다고 들었는데 오늘 이렇게 많은 분들이 참석해 주셔서 대단히 감사합니다. 자, 고려대학교 제3회 넥스트 인텔리전스 포럼에 참석해 주신 모든 분께 환영의 인사를 드립니다. 특히 오늘 뜻깊은 강연을 맡아주신 요하임 프랑크 박사님께 존경과 감사의 마음을 전합니다. 요하임 프랑크 박사님께서는 저온 전자현미경 관찰법 연구를 통해 생화학 분야의 새로운 시대를 열며 신약 개발에 필요한 중요한 단서를 얻을 수 있게 하였습니다. 이처럼 생물리학자로서 높은 연구 성과를 내신 요하임 프랑크 박사님은 2017년 노벨 화학상을 수상하셨습니다. 오늘 박사님께서 들려주실 단분자 초저온 현미경과 생체 물질의 시각화 강연은 고려대학교의 연구자와 학생들에게 매우 유익하고 시사점이 높은 강연이 될 것이라고 기대를 합니다. 이 자리에는 교수님들도 계시고 많은 연구자도 계시지만 또 아주 젊은 학생들도 많은데 궁금한 점도 많이 오늘 물어보시고 이 학문 세계가 어떤 식으로 지금 진행이 되는지 많이 참고를 하고 여러분들의 앞길에 도움이 되기를 바랍니다. 1905년 개교 이래 대한민국의 근대화, 산업화, 과학화, 세계화를 이끌어온 고려대학교는 2025년에 맞이할 개교 120주년을 계기로 인류의 미래사회에 공헌하는 세계 속의 명문대학으로 도약하고자 합니다. 고려대학교는 기후위기, 감염병 확산, 사회경제 양극화 등 인류 문명의 미래를 좌우할 문제를 연구하고 그 해결 방안을 제시할 수 있는 대학으로 성장할 것입니다. 세계 석학 강연 프로그램인 넥스트 인텔리전스 포럼은 개교 120주년 기념 사업의 일환이며 고려대학교가 세계 명문 연구기관이자 교육기관으로 더 크게 발전하겠다는 의지를 표현한 것입니다. 제3회 넥스트 인텔리전스 포럼은 특별히 고려대학교 이과대학 설립 70주년 기념 강연으로 열립니다. 고려대학교는 1953년 한국 전쟁의 험난한 상황에서도 기초과학의 중요성을 인식해서 수학과 물리학과, 화학과, 생물학과를 설치하였습니다. 그로부터 70년 동안 고려대학교 이과대학은 지속적으로 성장하여 세계 속의 명문이 되었고 대한민국 기초과학 발전에 혁혁한 공로를 세워왔습니다. 오늘 요하임 프랑크 박사님의 강연이 고려대학교 이과대학 교수님과 연구자들에게 더욱 각별한 의미를 갖게 되리라 기대합니다. 저도 오늘 끝까지 남아서 이 강연을 듣고 싶은데 저는 오후에 교우회 행사가 있어 바로 아마 인사 말씀 마치고 바로 출발하게 되어서 아주 아쉽게 생각을 합니다. 다시 한번 강연을 맡아주신 요하임 프랑크 박사님께 감사드리며 제3회 넥스트 인텔리전스 포럼에 오신 모든 분께 환영의 말씀을 드립니다. 아울러 넥스트 인텔리전스 포럼을 후원해 주신 정진혁 센트로이드 인베스트먼트 대표님께도 깊이 감사드립니다. 오늘의 포럼이 모두에게 소중하고 알찬 시간이 되길 바랍니다. 감사합니다.
이제 요하임 프랭크 박사님을 간단히 소개해 드리겠습니다. 프랑크 박사님은 극초원 전자 현미경을 이용하여 생체 분자의 3차원 구조를 원자 수준에서 관찰하는 길을 연 공로로 자크 두보시에, 리처드 핸더슨 교수와 함께 2017년 노벨 화학상을 수상하셨습니다. 교수님은 1975년에 하나의 단백질을 여러 각도에서 촬영한 저해상도의 여러 2차원 전자 현미경 이미지를 3차원 이미지로 결합하는 이론적인 전략을 발표하셨으며 1990년대 초에 세포 내 단백질을 생산하는 분자기구인 리보솜의 3차원 구조를 40옴스트롱의 해상도로 획득하는 데 성공하였습니다. 이런 이미지 처리법은 크라이오 EM 기술 개발에 기반이 됐으며 2013년 새로운 전자 탐지기가 도입되면서 마지막 기술적 장애물도 극복한 크라이오 EM은 막 단백질이나 거대 분자 복합체 같은 세포 내 다양한 분자의 구조를 원자 수준에서 포착할 수 있게 해주며 생화학의 새로운 지평을 열었습니다. 그럼 지금부터 프랑크 박사님의 강연을 듣도록 하겠습니다. 여러분, 프랑크 박사님을 큰 박수로 맞이해 주십시오. 자, Dr. Frank, yeah, please come forward. Yeah.
Dear President, esteemed faculty, it is a great pleasure to be here, and I thank the President for this very important invitation to this special event. I'd like you to learn about single particle cryo-EM. I was told that the audience has quite a mixed background in different fields, and so I decided to assume nothing and start from scratch and bring you to the level of what I want to get across. So we can discoveries in life sciences as we zoom in on the parts of an organism with increasing magnification. An organism, an organ, a tissue, cell, an organelle, and the lowest level is a molecule. And to visualize small objects, we can use microscopes. The light microscope goes down to 200 nanometers in resolution. Electron microscope goes down to 0.1 nanometer, and which is close to the size of an atom. And we have to first appreciate the universality of life processes on Earth. When we look at the organisms, we see an incredible variety. But if you go down to the level of the molecule, all of a sudden life appears to undergo exactly the same principles. And here's, for instance, in animal cell, first of all, the smallest unit that is somehow functioning is the cell, both in plants and animals and bacteria. And here's an example for the animal cell, which is very crowded with different organelles that have various functions that are used to maintain the metabolism. And so the cell is essentially packed with molecules, and life processes are created by the interactions between molecules. And to understand life in healthy or sick organisms, we need to know the structures of molecules. And medicine increasingly in the last decades is based on the knowledge of the molecular processes. And that's why we call, that's why there's a completely new field which is called molecular medicine. Medicine, in essence, is nowadays molecular medicine. It's all based on the understanding of structures. And there is David Gottsell from Scripps Institute, who spends his life drawing very intricate pictures of cells and what's contained in it. And from that, we get really a sense of the crowdedness, but also of the complexity. And I just show you some of these artistic and beautiful pictures. What we see in molecular detail are a lot of what we call molecular machines. These are molecules that are very complicated, and they interact with other molecules to make something happen, to build something up, or to analyze something, to destroy something in a very detailed way, and to transport molecules and so forth. This gives us an understanding that molecules don't just randomly interact and do something and then depart, but rather they do this in a very processive way. So processive molecular machines, that's a very important keyword. And what we really want to know is how all these processes work. Ideally, we want to make a movie, okay? Now this movie is really made by simulation, by computer simulation. And the question is, where do we get this information? How do we even conceive of this process and get such a detailed simulation? And so what do we need to know about molecules? They're composed of atoms. We need to know the atomic structure. If we talk about a molecule, we need to know the atomic structure, which means that we need to know the position of every atom in some coordinate system. And the word atom comes from atomos, which is a word that was used with Democritus and stands for uncuttable, indivisible. And Democritus already postulated that there was some unit, some smallest unit that cannot be divided anymore. And here's the periodic table of elements, and it shows you which of the elements are essential for humans. And you see there's only a small variety, and the most important ones are the hydrogen, carbon, nitrogen, oxygen, and then phosphorus and so forth. So the structure of a protein molecule is a table of the precise XYZ positions and identity of all of its atoms. And how do we determine the atomic structure? By X-ray crystallography or cryo-electron microscopy, or we simply say cryo-EM. Now, an example of a human protein is hemoglobin, which carries oxygen in our blood. And this contains about 10,000 atoms of hydrogen, carbon, nitrogen, oxygen, sulfur, plus 4 atoms of iron, which carries the oxygen. How are they organized? What keeps them together? Now, look at this display alone. The display of all atoms of hemoglobin. Does it tell you how it works? Not at all. We don't know how they interact, and we don't see any rhyme and reason. So we have to first understand that the protein structure is actually a linear structure. So there's a primary structure of amino acids, and this primary structure which is a polypeptide, folds into what's called a secondary structure. This can be alpha helix, it's a helical structure, or a beta sheet, where it sort of moves back and forth and so forth, and produces some kind of a firm structure. After this step of folding, we get the tertiary structure. All these different elements then collapse together, and then in the end, all the different tertiary structures arrange themselves into the quaternary structure, which is the actual protein. So this is a very complicated folding process taking place, and the folding process is mainly determined by the properties of these elements, of the amino acids. And properties, we mean hydrophobic, hydrophilic properties, electrostatic properties, and so forth. When we think of a protein, we can think about this display that is used for Christmas. And so it's really, everything is bunched together, but it is one linear chain. So the building blocks of proteins are the amino acids, and we always use a ball and stick model like this. Okay, so here is one example for an amino acid. And there are 20 different amino acids. They only differ in the side chain in this here. And everything else is always the same. So here are three examples of amino acids. And this reminds us again of this metaphor, okay? It's just different contexts here. And so the amino acids are strung together to form a long chain, which is called the polypeptide chain. And the chain folds up to become a protein, as I told you just before. And now how are the amino acids strung together? And they're strung together by this fascinating molecule, which is called the ribosome. It's a molecular machine that goes on in a very precise fashion. And there's a beginning and then an end. And it's a succession. There's a processivity involved, okay? And the ribosome gets its instructions from the genetic code, which resides on the messenger RNA. Messenger RNA, in itself, is a copy of a stretch of DNA, okay, of a gene. So here we have every atom representation that I showed you before. And if we use a ribbon diagram, it's a representation of the secondary elements. And they're all connected in a linear way because the protein is just one-dimensional stretch. So it is a vastly simplified representation. It shows the underlying 1D sequence, and it also shows the secondary structure elements, the alpha helices and beta sheets. And this is even more simplified, but it shows all the essential features of the hemoglobin. I just use the hemoglobin because it's a relatively simple molecule, even though it has 10,000 atoms. So now how do we find the positions of all atoms in a molecule? And this is the process called structure determination. And we also say we solve a structure. And these activities are part of structural biology. When I started with my research, there was no such a term. It didn't exist. Structural biology didn't even make sense as a notion. So we first get a 3D image either by using X-ray crystallography or cryo-electron microscopy. NMR, to some extent, is also used, but for rather small molecules. And then we use that three-dimensional image. It's really an image which has, for each element, for each voxel, it has a volume, in a three-dimensional coordinate system. It doesn't tell you anything about atoms. It's just a density distribution, or we call it a three-dimensional image. And from there, we come to a model of the atomic structure of the molecule. And that is a particular process that involves detective work. And I just show you the 3D image of the hemoglobin at 2.7 angstrom obtained by cryo-electron microscopy. Okay, so cryo-EM can do the same thing as X-ray crystallography, and here we have it. So here's the detective work. Atomic model building. We find which amino acid structure fits best into the electron density. Very often, or most of the time, we already know the sequence, so we know what to expect. And if we have already identified an amino acid at one position, then we know where to look for the next one. It must be in the close vicinity. Okay, so this is detective work that goes through this entire three-dimensional density distribution. So here is an example. The amino acids arginine, proline, and leucine appear at positions 89, 98, 91 in the sequence. And their atomic structures are depicted as ball and sticks models. And the bulges in the 3D image show perfect fit, okay? So if you didn't know that you would expect an arginine 89, a proline 90, and leucine at 91, you would not really know how to interpret this bulging structure because the resolution is not sufficient. But with that knowledge, and with the knowledge of what has already come before, we can build an atomic model safely. So just to talk about the methodologies here, X-ray crystallography. In X-ray crystallography, which has been used for decades in order to solve structures, an X-ray beam is shown onto a molecule which is in a crystal, and then we get diffracted rays, and we can interpret this pattern with some additional information which we get from phasing. And then we can get very high resolution structures. The problem is that many molecules cannot be induced to form highly ordered crystals. And if they're not highly ordered, then we get very low resolution and we cannot do this atomic interpretation, which really means that the universe of molecules falls apart into two sub-universes. One that is accessible by X-ray crystallography, and the other one is completely unknown, okay? And here's Max Perutz and John Kendrew, who received the 1962 Nobel Prize in Chemistry with a model of hemoglobin. And at the time, there were no fancy workstations. There was no way of even looking at them on images, even the projection images. Nothing was there. There were no workstations. And so they built this model by clamping sort of wires together and twisting them in a way that they inferred from computer outputs. So to summarize, many molecules cannot be crystallized, so X-ray crystallography cannot be used to determine their structures. And here I give you a list of structures where it was particularly difficult to get information. Receptors, heat receptors, mechanical receptors, hormones, signal molecules, and then among the ion channels, potassium, calcium, chloride, protons, and so forth. And then very large molecules, they are very large, especially molecular machines. They have floppy elements at the periphery, so they sort of resist being packed into a crystal. And so for many of these, there has been no information available except for just portions of them. So fortunately, the electron microscope comes to the rescue. Okay, so it came to the rescue early on. It forms projections of an object. And if we talk about transmission electron microscope, transmission means that electron goes through a transparent object. But to be transparent, the object must be very thin because the electrons get very rapidly absorbed. And what is the projection? So a projection is not a contour around some kind of a solid object, but a projection, the true projection is where you see an integrated density at each beam that goes through. So we don't really have a lot of illustrations like this in real life, but what comes to mind is the jellyfish. Okay, the jellyfish is transparent. We look all the way through, and they are sort of some gelatinous structures which have a little bit more density, and they're maybe a little colored and so forth. So so you look in one direction, you see a particular pattern which gets you information about every single element in the XY plane. And you can now step around and you get different views of that same object, of that same three-dimensional object. Here's an older generation electron microscope. So, in a very primitive way, we have to produce electrons at high speeds, and then be done by using a cathode where the electrons are emitted, and then an anode, so the electric field goes like this. The electron, which is negatively charged, is accelerated to the speed that is determined by the voltage difference. And typical voltages are in the range of 100,000 to 300,000 kilovolts. So the electron is really accelerated to close to the speed of light. And electrons behave in a very weird way in the magnetic field. They sort of spiral around around the curved magnetic field lines. So we can use magnets in order to redirect electrons, and we can actually use them to focus electrons. So an electron lens is formed by a magnet in this way. And the first one who thought about using electrons to form images was Ernst Ruska in 1931. He got a Nobel Prize in physics rather late in 1986. So this is the notebook sketch where everything was already figured out. And the amazing thing is that only a few years earlier, the duality of electrons, meaning that is a particle and at the same time a wave, was recognized by de Broglie. Before, this was a completely unknown concept. And so just a few years after that conceptual insight by de Broglie, we already have an instrument working with electrons as waves. That is unbelievable. But then the electron microscope was essentially dormant for a very long time, over 30 years in terms of molecular structure. It was mainly used for looking at tissues and rather very, very large morphology in slices, tissue slices and so forth. And So we can put next to each other the electron microscope and the light microscope, and and they are completely corresponding elements. We have the light source here, the electron gun here. We have an illumination system, which is here formed by magnetic lenses, here by a condensing lens, and here is the objective lens, the most important lens in the system. And then there comes a projection system that essentially magnifies the job that the objective lens has done. So we have a complete equivalence between these, and the only difference is that in one case we have enormously better resolution. And here it is, and what we need to remember is that there is a central column here which is on very high vacuum. It has to be on high vacuum because otherwise the electrons get stuck even in a small layer of air. So electron microscopy can be used to determine molecular structures. Projection images are formed at very high magnification. A typical number is 30,000. Sample must be very thin. Electrons are readily absorbed by matter. And electrons strongly, this is a very important point, electrons strongly damage the molecules, and there's the need for low dose. And a typical example typical value is 10 to 20 electrons per square angstrom. Unfortunately, with so few electrons per square angstrom, the image looks like salt and pepper. You don't see any clear image of a molecule. So what does it mean? What does it mean for the prospect of looking at molecules? It simply means that without averaging over many instances of a molecule, we cannot use electron microscopy at all. Okay? So radiation damage forces us to go down with the number of electrons per square angstrom. The result is that the image is so poor, we speak about shot noise, that we cannot recognize anything. So imaging by electrons and averaging always goes together. We cannot image a single molecule all by itself and expect to interpret it. And my PhD mentor in Munich was Walter Hoppe, and my PhD was between 1967 and 1970. Here he is with a microscope. At the time, that was sort of a high-end microscope from Siemens. And he was a very unique person because he had a background in X-ray crystallography. And for some reason, which I still don't understand, he decided to switch over to electron microscopy. He thought this is where the future was. And but what it meant for me is I grew up, in a sense, academically grew up in an environment in which all these concepts, like Fourier concepts and the Ewald sphere and everything was there. And so the concepts were expressed in a highly sophisticated way. Of course, Hoppe thought about electron microscopy in terms of all the tools that were already available in X-ray crystallography. And he was optimistic. He thought you could just rewrite the programs a little bit and then, now you got the phases and everything is hunky-dory. But it was really not so. I was the first one who used image processing in order to try to make sense of electron micrographs. And the discoveries that I made were horrific. It was terrible. The noise was overwhelming. It was very difficult to extract the signal. So an entire branches that had never been considered there, branches of science had to be consulted in order to make sense. For instance, signal processing of electrical engineering and statistical optics. These were all fields that had a lot of contributions, but they needed to be put to use in these interpretations. So that's where I was. I was forced into this interface there, and it didn't feel very comfortable there. So here's a later version in year 2000, just to show you how the electron microscope itself grew up. The matter of 3D reconstruction. How do we get from a bunch of projection images to a 3D image? Well, there's this stupendous paper by de Rossi and Klug in 1968. This was absolutely pioneering work. It was the first time that 3D reconstruction was done with a computer. There were optical contraptions before, but it was the application of theorems that were developed in 1905 by Johannes Radon. Okay, what it meant was, and they applied this to bacterial phage tails, which have a helical structure. According to Radon's theorem, we can make a 3D reconstruction of an object in the following way. We image the object from different directions and then record the projections. And according to that theorem, these projection images form central sections in the three-dimensional Fourier transform of the object. And that suggests the recipe that we get views from all different directions and then sort of gradually fill the three-dimensional Fourier space and then do some kind of interpolation here so that we can get a complete coverage of 3D Fourier transform. And then we do a back transform and we recover the object. So this was sort of the outline. And it's much simplified in this way, in this case, because of the presence of symmetries. Presence of symmetries, like helical symmetries, means that if you have a measurement at one place in Fourier space, you immediately have measures at other places in Fourier space as well. So another example here is a 3D reconstruction of viruses with icosahedral symmetry by Tony Crowther, and later on, 3D reconstruction of the two-dimensional crystal of purple membrane protein by Nigel Unwin and Richard Henderson. So now, what do these three applications have in common? They all have in common that the molecule are not just looked at by itself, not as a molecule, but always in a crystalline formation. In one case, a helix, in the other case, a spherical virus with high symmetry, and here in a plane. What did it mean for the computation? Well, it meant that the programs that were developed were very specialized for particular geometries, okay? So each time you had a particular symmetry, you had to redo everything again, okay? And then also very clear from the beginning is that there are only few molecules that actually form beautiful crystals. And what about the other ones? And the natural question is, do we need crystals? Can't we just tilt the molecules to get all the views we need? Well, that was the idea that my mentor had. He thought we put the molecules simply on a grid and then tilt the entire grid around, and then we get a 3D structure of the grid along with 3D structures of the molecules. We just carve them out and look at them. So the problem with this so-called tomography approach is that the molecules get damaged by the beam. And since we have the same molecules tilted and radiated over and over again, we get an accumulation of those, much more than the molecule can withstand. So it's a bad idea, because we get a beautiful 3D reconstruction of a molecule that is completely damaged, okay? So it's completely useless. Okay, so there came this idea. What about using single particle reconstruction? Make use of the fact that molecules occur in millions of copies, all in random orientations. So they exist in so many copies, so averaging can be used over low dose exposures, and they are in random orientations, which means we don't even have to tilt them, okay? They're already tilted. They are already doing us the favor of being tilted. So this is the idea of a single particle approach. Here's the view. The molecules are simply in solution. We managed to get them onto the grid. And at the time, there was no cryo-EM. There were no cryo methods. All I had was a negative staining. Sort of stain was being used in order to produce contrast. So it's a nice idea, but then how are we going to do this? The molecules are oriented by themselves, but we don't know the angles, okay? So I have only postponed the problem to another step, and that step is the computational step. So a lot of work had to go into making the necessary programs. And that's why I'm saying the devil is in the detail. So we had to make programs that dealt with all these different issues. And the finding projection angles is actually the biggest one, but then also sorting and classifying images so that we don't have to rely on subjective interpretations is an important one. And this is what I worked on 1975 to 1986, and that is sort of my share of the Nobel Prize. These were very intensive years where we managed to do this. And this is the first proof of concept where I showed that with these techniques we get beautiful two-dimensional averages of these noisy micrographs. And then in 1987, there was the first single particle 3D reconstruction of the large subunit of the E. coli ribosome. I told you about the ribosome before, and this is here now, the large subunit for the first time seen in 3 dimensions and presented as this polystyrene model, because there was no way of presenting it in the computer. But it still relied on negative staining. And this is another way of presenting it, making contour plots and then stacking them up with the right kind of Z distance in a wooden box, and then displaying them on a light box. Okay, this is a very heavy thing. It's 11 pound. And then came the other revolution, which was the frozen hydrated specimen preparation, where Jacques Dubochet played this important role. And the first paper relating to this was published in 1981. So now, from now on, the specimen, the molecules that are in solution, a drop of these were put onto the grid, and the excess liquid is blotted away, and then the grid with this thin layer of molecules is quickly dropped into liquid ethane on liquid nitrogen temperature. And this particular, when the plunging is very fast, then the ice that is formed is not the crystalline form, but it's vitreous. It's like glass-like. And under these conditions, the molecules are preserved in their native structure. And from that moment on, we talked about cryo-electron microscopy. Before, it was just single particle electron microscopy. Now, these two techniques joined together, the cryo techniques and the single particle technique. And these were wonderful years because we, for the first time, we found many beautiful structures of molecules. They were still at low resolution, but still, they were the first ones of these molecules because they could not be crystallized. It's the ribosome, hemocyanin of many forms, this is just one part, and the calcium release channel. And this was, in these years, we also found the first evidence of motion. And here is, in 2000, we discovered that when the factor EFG binds to the ribosome makes a very large movement by 7 or between 7 and 10 degrees. And this intersubunit motion is required for translocation of the mRNA from one codon to the next through the ribosome. So this even happens in the absence of EFG in solution. At a normal temperature, many molecules can just equilibrate between different states. Okay, the thermal energy is sufficient to bring them from one state to the other. So what happens when molecules are in thermal equilibrium? What kind of pictures do we get? So the multiple states coexist at that temperature, and they would all be observable, but they would all be intermixed in the image. So I just show you a simulation of what's going on. Now this is a microscope grid, 3 millimeter microscope grid, and it's covered with carbon, and the carbon has holes. You can see the indication of these little holes there. Okay, now we see these grid squares more closely, and we see all the holes. Okay, now here we look at a single hole, and now we see the molecules there, and just imagine they all change between different states. You know, they do this intersubunit rotation. And then comes the big freeze, okay? They're plunged, and whatever state they are in, it is now frozen, okay? Which means that we have a multiplicity of states there. For simplicity, we say they're just two, okay? This way and that way. We might not see the intermediates. So then this would be the computer image, and you see it looks like salt and pepper because of the low exposure that we have to use. And so now we are in the computer, the image is already in the computer, and then we have an automated system that just looks for these masses and puts windows around and puts them into a gallery. Okay, so now we have a gallery, and now we can shuffle data around and average them and classify them. So obviously, since we have a mixture between these two different states, we need an automated way of classifying them and sorting them out according to which structure they come from. That seems almost hopeless, okay? Because we don't know the orientations, we don't know the classes they come from. The images are very noisy, but it's still manageable with maximum likelihood methods. So they are very clever programs that we can use, which are doing this kind of sorting. This is just an example for some project when we have a complete pool coming from the same sample, and then we discover all these different categories, okay? We can sort them out by three-dimensional classification. And then we get an inventory of everything that is in the sample. And that gives us some kind of a story. You know, we can interconnect these structures using information from existing literature. So this is an example where we, at the time where we still had low resolution, because the recording system in the electron microscope was not perfected, and the microscopes themselves were not as stable as they are now, but we were already able to use these methods to look at different states of a system of the molecular machine, like a ribosome, and look at different stages. So we were able to figure out the elongation cycle. The ribosome has to go through one of these cycles for each amino acid that is being added on. And we were able to find the structures at each of these different steps of the elongation cycle. And then came the resolution revolution. In 2012, the first commercial direct electron detection cameras became available. Before that, the best way of recording images were on photographic film. And photographic film actually is very lousy. It's not only inconvenient because you have to operate in the dark, you have to develop and then scan the whole thing into the computer. But the quality of recording is also very bad. So these cameras were a real revolution. They're much superior to recording on film, and part of that resolution was also that the electron microscope became fully integrated and automated with the computer. Now, all of a sudden, we had such an amount of control over taking images. We could essentially take images in an automated way after giving the computer the marching order on the grid. And that's how the new era of single particle cryo-EM came about. Yeah. So to give you a sense of this resolution revolution, it's very often it is said, well, you know, I mean, we had some single particle structures already before 2012 and so forth. Well, but the point is the restriction by that was posed by the poor recording system, you can think of this as something very trivial. Everything else worked. You know, the math worked, the computation worked, the electron microscope worked. And there's this poor little, you know, thing. So the information was there, the information arrived there, but it was not recorded correctly, okay, in a very poor way. So this is really what happened. All of a sudden that information became available and it was high resolution, and then people took off with it. So many papers appeared after that. And here's the, you know, if you want to know what is happening, the best cameras before were CCD cameras, and they had a very poor performance because they converted electrons to light and then light back to electrons again. And here there's an immediate transduction of the information. And this is how crisp images look now. This are images of ribosomes. And you might have seen this before, but this is the scale of microscopes now. They're completely self-contained and inside of boxes in order to get them thermally insulated. And here you have a look inside. They're packed almost like an animal cell. So with computational, with computer elements and elements of electron optics. So this is now the workflow of structure determination by cryo-EM, sample preparation at cryo-EM, grid setup, cryo-EM imaging, data collection, data processing. You reconstruct a map, and then we do model building, and we reconstruct a map, and then we do model building, and we get a structural analysis and try to make sense of what we observe. And just a few years after this camera came out, we were able to get an atomic structure of our favorite molecule of the ribosome at 2.5 angstrom resolution. And here you see that inside, if you make a cut through the ribosome, you see that the resolution even reaches 2.3 angstroms inside. So we can now look at amino acids and see the detailed structures at that resolution. And here's an example of a base pairing that goes on in the ribosome. And we see individual magnesium atoms. We can spot water molecules, and it's in an entirely new world that we're in right now. And I told you that we can retrieve different structures that coexist in a sample, and I call this story in a sample, and here are two examples of this kind of phenomenon. There in two papers, we show how the ribosome as a molecular machine coexists in the different states. And then, I've always concentrated on the ribosome because that was sort of the demo particle for doing the development of the method, but then it became really an integral part of the work, of the biology work. But then after I came to Columbia and brought a high-resolution instrument with me, I was approached by many people to do collaborations, and so I got into other molecules and receptors, ion channels, and so forth. And here's the calcium release channel and the AMPA receptor. And nowadays, if you just go on Google and you can type in cryo-EM of molecules, you can get pages and pages of this here. So it is really everywhere. And in many cases, X-ray crystallographers have left X-ray crystallography and became part of the cryo-EM community. So here are sort of the individual steps, but I cannot go into detail now. But just to show you the timeline, there's first the single particle techniques, and then came the cryo-EM revolution in 1987. Then came the resolution revolution right here, 2012, and then this was from a review article that I wrote for Nature Protocols, and I have extended this with this slide here because what happened was that, in my opinion, 2012, the cameras arrived just in time to make us able to cope with the various terrible diseases, with the MERS, Zika virus, Ebola, dengue, and then SARS-CoV-2. In each of these fields, for each of these viruses, cryo-EM, single-particle cryo-EM has been crucial in making discoveries and promoting the field, and in the case of SARS-CoV-2, contributed to the rapid development of the mRNA vaccination. And just to drive this home, just 2 weeks after receiving a genome sequence from Chinese researchers, the people here around Jason McLellan had already designed and produced samples of the stabilized spike protein, and it took 12 days to reconstruct the 3D atomic scale map. And it was published in Science, I think at the beginning of March. So this just 2 months after he received the genome sequence, he already had this structure. And I just want to emphasize that cryo-EM accelerates drug discovery by delivering previously intractable structural insights. And there are examples that I found in a review article. The drug is binding to GPCRs, to ion channels, and selective sodium ion channel blockers. And then cryo-EM structures reveal new potent selective inhibitor of parasite enzyme. I'll just show you this in the next slide. And then reveals an allosteric site for enhanced druggability of ATP citrate lyase, insulin binding, viral epitope, and then the stalling ribosome by small molecules. And this is an example of an inhibitor of parasite enzyme that was done by analyzing the cryo-EM structure in detail, and it proved to be possible and proved to be a potent drug. So this is just one of many, many examples that I can bring to show you that cryo-EM is an essential tool for the pharma industry of the future. In conclusion, single-particle cryo-EM is a powerful method to visualize biological molecules in their native states. No crystals are required, and we see the functional relevance of the structures obtained. Multiple structures can be visualized from the same sample. And novel methods, time-resolved cryo-EM and mapping of states in a continuum. I have not talked about this, and I would be running out of time. But this is an image that I need to show because it shows me with Carol, who is present in the audience, just a very short time after I received a call from the Nobel Foundation. This is an opportunity to thank Carol for being with me for all these different difficult years. And that brings me to that picture. And we see the box again. This time I was asked by the Nobel Museum to bring something that was related to my research, as every Nobel laureate is. And I thought the most appropriate thing is to bring this box that had the first single particle 3D reconstruction. And here we are in 2021 with a slightly smaller group at the Washington Bridge, George Washington Bridge, at the little lighthouse. And the significance of this picture is that it's the first picture where the group came together again after COVID. And we decided to still be on the cautious side and meet outside. Okay, thanks very much for your.
프랭크 박사님의 좋은 강연에 다시 한번 감사드리고요. 자, 지금부터 문답 시간을 갖도록 하겠습니다. 우리말이나 영어 어느 말로 하셔도 됩니다. 우리말로 질문을 하시는 경우 우리 고려대학교 화학과 심상희 교수님께서 그 통역을 하여 주실 겁니다. 자, 질문을 하시는 분은 간단하게 본인의 소개를 좀 해주시면 감사하겠습니다.
Hi, my name is Art Cho. I'm a professor in bioinformatics at this university. Thank you very much, Professor Frank, for a very educational and informative talk. Well, by profession, I'm a computational biophysicist, and I'm sure you already know that in my field there has been a great stride in protein structure prediction methods with the introduction of AI technologies. Now, some actually predict. I don't agree with that, but they believe that it will eventually put experimentalists out of job. But I think there must be some way that the experimental methods are collaborated with the theoretical computational methods. And I would like to hear your opinion on the future of the role of the theoretical and computational work with the experimental work in structural biology in general.
Yeah, the opinion that you quoted is total foolishness. It is, of course, the experiment that is really the touchstone of science. And if we get simulations, then these are just simulations. They're simulations on existing knowledge. The existing knowledge is limited. Even though we've been doing structural biology for decades, a very large part of the universe of structures is unexplored. That part of the universe is unavailable for training of these smart computers, these smart systems. So there's essentially there's a principal side involved and there's a practical side involved. The principal side is it's not science if you just look at simulations. I wouldn't want to go to a treatment where the drugs are developed on the basis of AlphaFold predictions. I wouldn't want to go to a doctor and get treated on that basis. I want to have experimental results that actually confirm these predictions. And there's no escape from this. So it's really utter foolishness. I was astounded by the opinions that were expressed. I think they betray a fundamental misunderstanding of what science is.
Okay.
Thank you, very nice talk, and thank you for what introduced to the human being. My question is, up to date, we can't get the 3D structure of many proteins or many receptor, although the cryo-EM not need the crystal structure. And in this case, we need the homology modeling to expect maybe the active site of certain protein to discover a new drug. So all the receptor or all the protein in our body, we can get the 3D structure. So what's the problem here? If the cryo-EM don't need the crystal, yeah?
If the cryo-EM doesn't what?
Yeah. So a cryo-EM doesn't need the crystal to get the 3D dimensional
Yes.
structure. So up to date, there is many proteins or receptor, we can get the structure of this protein. So
Yes.
what's the problem here? The problem in the protein itself or the problem in the technique or?
Oh, yeah, okay. Are there any remaining problems?
Yeah.
Okay. Well, a lot after the perfection of the data analysis itself for well-behaved proteins or protein RNA complexes, a lot of attention is focused now on producing well-behaved samples. Often you find that proteins aggregate and then they just don't present themselves as single particles. And there are some very important assumptions made in this entire processing. And one of them is that the molecules are not in an aggregated form. So there's a lot of energy is now being put into optimizing sample preparation. And there are robots being developed to do this very reproducibly and efficiently. But a lot has to be done still.
Okay, question. Mike, please.
Thank you for your great lecture. So I'm the second grade student from the College of Medicine, and I have some sort of young questions. So the question is, why did you study or research this field? Like, is it because it's necessary or just simple interest or something else?
Yeah, it's really, it was a pretty arbitrary development. I mean, I can't tell you that I just had this goal in mind all the time. There were opportunities to go in this way. And my earliest interest was really in electron microscopy and how to perfect it and how to use it for molecular structure. But the idea of single particle really came all of a sudden in a particular situation where other solutions obviously didn't work, and so many people, many students who ask me, how did you get there and so forth. It's always very much by accidents. There's an accumulation of accidents, and if there's one trait that is very important, it is to recognize opportunities on the wayside. All of a sudden there are opportunities, there's some kind of insights that come from unexpected directions.
오케이. 우리 쭉.
I am ex-faculty at the physics department. As a physicist, I'm always amazed that the chemistry people are doing much complicated system building well. As a magnetic resonance major, I am wondering what is the comparison between your method and magnetic resonance method of determining molecular structure.
I'm sorry, I found this difficult to understand. But,
NMR method
yes,
was also applied to determine the molecular structure of biological systems
yes.
much earlier in the 2010 or something. And your method is recent one, using electrons through the material. So I am wondering, what is the difference between these two methods to determining the structure?
NMR is still rather restricted to relatively small molecules. And electron microscopy has no such restrictions. Electron microscopy is particularly powerful for very large molecules, where NMR can really make very little contribution. And so I don't know enough about NMR, because I realize there have been new developments that go toward larger molecules. But you can see in the depositions in the protein database that NMR contributions have really leveled off, whereas cryo-EM contributions really have an exponential rise.
저 대단히 미안하지만 질문 딱 하나만 받고 이제 그치도록 하겠습니다. 손을 제일 높이 드는 학생이 여기.
네. 한국어로 해도 되나요? 네. 안녕하세요. 저는 수학과 석사 과정 재학 중인 학생이고요. 궁금한 점이 이제 이미지 리컨스트럭션에 대한 얘기가 있었는데, 이게 일반적으로 이제 휴먼 스케일에서 생각을 해보면은 그런 것들을 실제 이미지를 가지고 3D 이미지를 구상하는 그런 모델들이 많이 있는데, 여기 있는 것처럼 나노 스케일이나 그런 마이크로 스케일에서 그런 것들을 할 때 그런 스트럭션이 얼마나 퍼포먼스가 좋은지 그런 거를 체크를 어떤 방식으로 하는지가 조금 궁금합니다.
So the question is about the accuracy of the reconstructed image afterward. How do you check it?
Yes, so the question is essentially about validation. What's the accuracy, and how do we make sure that our results are correct? Well, it's in many investigations we need to have these kinds of cross checks, and I give you an example. The validation of structures. I told you the density map itself doesn't tell you where the atoms are. We need to do atomic modeling, so we need to find where the amino acids are sitting, and this is detective work, which is sometimes assisted by software. And so in the end, we have an atomic structure, but then the question is it correct? Well, there is an internal check for correctness, because atoms cannot be superposed. They are expected to be only a certain distance away from each other and not too close, and so forth. So there are many and you can realize what a gigantic job it is to validate this, to make this happen, to find out that everything is perfect. So what is happening currently is when you have an atomic model, you deposit it into the protein data bank, which is a precondition for publishing a paper. And so then the protein data bank makes a validation check. It checks out whether the chemistry the stereochemistry is correct of this entire model. And they send you a validation check. So it's a very large table of possible problems, real problems and possible problems and so on. This validation check needs to go together with the manuscript draft to the journal. They don't accept the paper before getting the validation check as well as the deposition of the structure. So this is one of the validations. There are others when you, for instance, you make functional predictions of the importance of a certain residue, then you can validate this. You can validate this by making a mutation there. So you go back to the bench, make a mutation there, and then demonstrate that the function doesn't work anymore. So these kinds of things are done all the time.
자, 아쉽지만 질문 시간은 이것으로 마치도록 하고요. 강연의 마지막 순서로 책 서명식을 갖도록 하겠습니다. Dr. Frank, we are going to have a book signing ceremony, so the book will be right here. 이 책은 프랑크 박사님의 논문집으로 고려대학교 과학도서관에 일정 기간 동안 특별 전시된 후에 대출이 가능하게 될 것입니다. 책의 제목은 Single Particle Cryo-Electron Microscopy입니다. 지금 뭐라고 쓰시는지 저도 굉장히 궁금하고요. 나중에 궁금해서 책을 빌려오시는 분은 꼭 전문을 읽어보시기 바랍니다. 자, 이상으로 강연을 마치고요. 우리 프랑크 박사님께 다시 한번 큰 박수로. 예.